Greetings, and welcome to Meiosis with Steve. Let's get started. Chromosomes are tightly coiled pieces of DNA. They consist of chromatids, members of the pair, and the centromere, the region where they are joined. A haploid cell refers to a cell with only one copy of each of its chromosomes. This is notated by the letter N. A diploid cell refers to a cell with two copies of each of its chromosomes. This is notated by 2N. The purpose of mitosis is to produce new cells for growth. It is also used to replace old or damaged cells. One diploid somatic cell is used to create two identical diploid somatic cells. The purpose of meiosis is to produce new gametes from sexual reproduction. One diploid germ cell is used to create 4 haploid gametes over the span of 2 cell divisions. Reduction refers to meiosis 1. In this process, during prophase, chromatin condenses into chromosomes and homologous pairs pair up. This is where crossing over occurs. Next is metaphase, where the homologous pairs line up across the metaphase plate. During anaphase, the homologous cells separate and are pulled to opposite ends of the cell. During telophase and cytokinesis, the poles separate into 2 cells. Division refers to meiosis 2. In this process, the same steps, prophase, metaphase, anaphase, telophase, and cytokinesis occur, except now there are no homologous pairs within individual cells. Synapsis and independent assortment does not occur again. Sister chromatids separate at the centromere this time, and 4 haploid cells are produced from the original cell. Synapsis, also known as crossing over, it occurs during prophase 1 and is the process of homologous non-sister chromatids exchanging genetic material. Independent assortment. This occurs during metaphase 1 and is the principle that alleles for separate traits are passed independently of one another. However, due to genetic linkage, which is the tendency of traits close together on a chromosome to be inherited together, there are some exceptions to the rule. Random fertilization. This is the principle that the collection of genes within a gamete do not affect the chance of producing a zygote. Random fertilization, independent assortment, and synapsis are all things that increase genetic variation and makes each person completely unique. That's all. Now, are you a wiener or are you a winner?